Welcome to the technical demonstration of the LifeWatch Tesseract Virtual Research Environment, IJI NIS workflows. We are going to start exploring the system by logging in. We type in our lifewatch.eu user and our password. Once we log into the system, we can see the main layout with the user menu, the username, and the link to log out of the system. And on the left, we have a list of all the workflows that this user has access to. This user has previously requested access to these workflows. Even though the workflows can consist of many tools, all of them have two common elements. One is the dashboard, where you can see which experiments are currently running and their state. The other is the Run a New Workflow button, where you can execute a new experiment. The workflow that is going to be shown is named Functional Biogeography of Invaders, the case of two widely distributed omnivorous crustaceans. Which are the scientific questions to be answered? 1. Can the ecological impact of invasive species be predicted using satellite-derived bioclimatic variables? 2. Can the ecological impact of invasive species be predicted in a changing climatic scenario? 3. Do post-invasion shifts occur in the trophic position of invasive crustacean species? 4. If any, which are the main bioclimatic drivers of these shifts, and will their contribution change under different climate change scenarios? And finally, is there data currently available to solve the previous 4 questions? To run a crustacean's functional biography workflow, we have to go to the left menu and click on Run New Workflow. The first thing presented to the user is a static diagram of all the necessary steps that the workflow needs to execute in order to obtain the predictive trophic map, and which files this workflow requires as input data. The first piece of information the user is requested to provide is a conceptual name for the new workflow. In this way, the user can identify and remember each workflow more easily. For instance, Friday morning experiment. The next step is to introduce the biological data on CN stable isotope, selecting the input file from our computer. After that, we select the trophic position data. Once the user has specified all the data, we can start executing a new workflow. To do so, the system will first upload the selected data and register a new workflow. Once this is completed successfully, the system will allow the user to run a new workflow. This would restart the demonstrated process all over, or go to the dashboard. If the user presses the dashboard button, the system will display an overview of all the current workflows for this user. If all is pressed, the system will show all the workflows. Pending will show all the workflows that have been submitted to the system but not yet executed. Started will show the workflows that are currently being executed. Failure will show any workflows that have had an error during execution. Revoked will show the workflows that the user has decided to stop executing. Success will show all the workflows that have been completed and whose results can therefore be downloaded. This workflow has an additional tool. As we can see from the workflow diagram, one of the steps is the geographical validation. This is a user interface which allows the user to manually validate all the data that has been previously filtered by the algorithm of this workflow. You can see how this tool works by pressing on the geographical validation button on the left bar. The geographical validation tool is designed to efficiently visualize all the samples that have been input by the user for this workflow, and to filter in or out all the samples that the user considers valid or invalid. To do so, the user can visualize all the samples displayed on a map. At the bottom, there is a table containing all the information of the samples, such as the data that was collected and a description of it. To help the user with the visualization of the samples, this tool can be made full screen. Also, the different sections can be expanded and collapsed. The map has a built-in functionality to allow the user to zoom in a specific area using the mouse. The user can mark a specific sample as valid or invalid using different tools. The first method is done by selecting one or multiple samples to validate. The toolbar on the right can then be used to specify whether or not these selected samples are valid. The second option involves exploring the table of samples. A sample can be located on the map by clicking on the Select on Map button. The user can mark whether a sample is valid by clicking on the corresponding checkbox. Once all the manual filtering has been completed and the user has made sure that all the data has been reviewed, the Continue the Workflow Execution button at the top of the page can be clicked on. The next workflow that is going to be shown is named Module 1, Atlantis Altissima Mapping Algorithm, and can be found in the left menu. Atlantis altissima is an invasive deciduous species. This means that it has an annual life cycle, with a biomass which peaks in spring and enters senescence in winter. The algorithm consists of two cascading stages. The first stage utilizes a multi-class land cover map to extract the deciduous vegetation layer, where its classification is based on either a data-driven approach or a knowledge-driven approach, depending on the availability of ground truth data for the training of a classifier. The second stage applies a binary mapping algorithm to Atlantis altissima versus other deciduous vegetation. The results from this first module will be connected to Module 2, Optimal Control Model Workflow. As demonstrated previously, when the user goes to Run New Workflow, the first thing that we see is a static diagram of what this workflow consists of. It shows all the input information which is required to run the workflow and all the steps required to produce downloadable output. As demonstrated before, to run a new Atlantis altissima mapping workflow, we should provide it with a workflow name so that it can be identified easily. For instance, evening experiment. Then, the user will be requested to provide all the satellite seasonal images. Next, the user will be requested to provide the information which was collected from the spring and autumn of the current year. Finally, the user will be able to create a new workflow. The system will then load all the files and start running a new workflow. As demonstrated before, we can see an overview of all the workflows that are running on the dashboard. We can filter those by state, and finally, for the ones that have been completed successfully, we can download the output. During the development process of all these tools, reusability has been taken into account. This allows us to reuse the components that we already have and create tools of the future. For instance, this is an improved version of the Workflow Studio. Here, scientists will be able to select different components from various workflows and thus create their own workflows.